Yes, so we have talked about the first genetic material, the DNA, and now we go into the second genetic material, the RNA. What is exactly the RNA all about? Let's analyze this beautiful concept. RNA in us eukaryotes helps in protein synthesis. Beautiful logic. What a concept it is. Let us first understand everything about what is the structure of an RNA and how it helps in protein synthesis to take place. So, RNA, the RNA is found in the nucleus as well as in the cytoplasm. DNA was found only in the nucleus. RNA is produced in the nucleus with the help of DNA, and then it goes into the cytoplasm. And in the cytoplasm, RNAs help in protein synthesis. There are two main types of RNA: genetic RNA and non genetic RNA. The genetic RNA acts as the genetic material of some viruses. Some viruses do not have DNA as the genetic material. The RNA itself acts as the genetic material. Which viruses are these? You have TMV, tobacco mosaic virus. And then you have bacteriophages like QB bacteriophage. QB bacteriophage, the deadly HIV virus, human immunodeficiency virus causing AIDS, has RNA as genetic material. Hepatitis A virus has got RNA as genetic material. And nowadays, the most commonly talked about virus, the deadly coronavirus itself, the SARS CoV 19 virus, has RNA as the genetic material. But except for some viruses, all other organisms have got DNA as genetic material, including us humans. We have DNA as genetic material, and we have non genetic RNAs. The non genetic RNAs function for protein synthesis. So, in us, our genetic material is DNA, RNAs help in protein synthesis. What is the structure of an RNA? RNA looks very much like the DNA, but DNA is double stranded, RNA is single stranded. And RNA is a single stranded structure. It may be simple or straight or may be folded upon itself. So RNA straight हो सकता है या it can be in the form of folds. It can be even folded, and that is RNA straight or folded. And chemically, it's a polynucleotide molecule. So, like DNA, RNA is also a polynucleotide, consists of a ribose sugar. DNA had deoxyribose sugar, C5H10O4. RNA has got ribose sugar, C5H10O5, which means if you remember, at the second carbon, it has got a hydroxyl instead of a hydrogen. So that is the one difference. The sugar is the ribose sugar. And the nitrogen bases, along with the ribose sugar, phosphates are in the form of phosphoric acid. That is same, H3PO4. And nitrogen bases, purines are adenine and guanine, as in DNA. RNA में pyrimidines are uracil and cytosine. In RNA, thymine is replaced by uracil. And the strand of RNA is a long chain of nucleotides joined by phosphodiester bond. So that is same as DNA. RNA is having in it phosphodiester bonds. So connection is done by phosphodiester bonds, exactly like DNA. And RNA is having four different types of nucleotide adenine, guanine, uracil, and cytosine, and it has got two ends, 3,5 end and 3 C3 end, C5 end and C3 end, exactly as in DNA. At the fifth carbon, there is a free phosphate. At the third carbon, there is a free hydroxyl. At the fifth carbon, there is a free phosphate that is called as the C5 end. At the third carbon, there is a free hydroxyl that is called as the C3 end. That is same. The RNA molecule, if it is coiled or folded, base pairing takes place. Adenine pairs with uracil or uracil pairs with adenine by two hydrogen bonds. And guanine pairs with cytosine or cytosine pairs with guanine by three hydrogen bond. This is a linear RNA. But samjho the RNA is folded and it has got folds. Then adenine pairs with uracil, uracil pairs with adenine, guanine pairs with cytosine, cytosine pairs with guanine, uracil pairs with adenine, adenine pairs with uracil, guanine pairs with cytosine, cytosine pairs with guanine, and so on. So it has got A pair with U by double hydrogen bond, G pair with C by triple hydrogen bond. And in the RNA molecule, base pairing is not fixed and definitive. तो उसमें purine pyrimidine ratio may or may not be necessarily 1:1. In DNA, there is a definitive purine pyrimidine ratio of 1:1. Chargaff's rule is always true in case of DNA. RNA, the purine pyrimidine ratio may or may not be 1:1. The Chargaff's rule is not necessarily true in case of RNA. Which are the types of non-genetic RNAs? There are three types of non-genetic RNA. They play an important role in protein synthesis, and these are mRNA, rRNA, and tRNA. So, mRNA, rRNA, and tRNA are the three types of RNAs. All three types of RNAs are produced on the DNA template by a process called as transcription. तो transcription से mRNA, rRNA, and tRNAs are produced. With the help of DNA, you have mRNA, rRNA, tRNA produced by a process called as transcription. Let's study these RNAs. First, we have mRNA, also called messenger RNA. Then we will talk about rRNAs, which is called ribosomal RNA. And then we will talk about tRNA, which is called transfer RNA. So we have messenger RNA, ribosomal RNA, and transfer RNAs. What is the mRNA all about? mRNA आपके डायग्राम में नोट्स में एक छोटा सा वेरिएशन करना है. By mistake, here it is given C3 end and here it is given C5 end. Cancel that and make this end as C5 end and this end as C3 end. So this is the C5 end and this is the C3 end. And this mRNA is a single, simple and straight without any fold. mRNA में कोई fold नहीं होता है. mRNA is without any folds. It is a linear molecule. rRNAs and tRNAs, rRNA and tRNA are folded. rRNA and tRNAs are folded. This is tRNA. It is folded. rRNAs are folded. mRNA is without any folds. So as we're seeing, mRNA is linear without any folds. The mRNA carries message for protein synthesis from DNA to ribosomes. mRNA carries a message from the DNA to the ribosomes. Because it carries message for protein synthesis, it is rightly called as the messenger RNA. It is about 3 to 5% of the total RNAs. If you count at random any 100 RNAs inside a cell, then only 3 to 5% are mRNAs. They are the least in number. सबसे कम नंबर किसका होता है? mRNA. Only 3 to 5 out of 100 RNAs, 3 to 5% are mRNAs. Least number of RNAs, neat point of view. Which is the least number of RNAs among mRNA, rRNA, tRNA? mRNA has the least number of RNAs. It has a molecular weight of up to 5 lakh Daltons. 5 lakh Daltons का molecular weight होता है. It has two ends, C5 end and C3 end. It is made in the nucleus by a process called as transcription of the DNA. It is made by a process called as transcription of the DNA. So let us see what is transcription. Sir, suppose I pick up a cell, say beta cell of islets of Langerhans of pancreas. This is the beta cell of islets of Langerhans of pancreas. And cell has in it nucleus, nucleus has in it DNA, segments of DNA are called as genes. So suppose this is gene number 11,111. If you remember, I told you there are 30,000 genes. तो हमने एक hypothetical gene लिया है, gene number 11,111. So look at this carefully. This is the DNA, segments of which are called as genes, and ये है gene number 11,111. यहाँ हेलिकेज एंजाइम आता है, याद रखना, हेलिकेज, and will start coming and breaking the hydrogen bonds between the two DNA strands. तो हेलिकेज आके हाइड्रोजन बॉन्ड्स तोड़ता है and the two DNA strands will uncoil. Sounds familiar? This is like DNA replication. But याद है DNA replication में first the DNA endonuclease had broken it into different fragments. And after endonuclease breaks, then helicase removes the hydrogen bonds. Yahan, we are not replicating the DNA. So there is no action of endonuclease. Endonuclease does not break the hydrogen bonds. Phosphorodiester bonds. Endonuclease से phosphodiester bonds टूटते नहीं है. Directly, helicase enzyme comes and breaks hydrogen bonds. तो AU, GC, इनके बीच में जो hydrogen bonds है, helicase breaks the hydrogen bonds. And the two DNA strands will uncoil. So it was looking like this. Gene number 11,111. Helicase breaks hydrogen bonds and the two DNA strands uncoil. So it will now look like this. So once again, so we have two uncoiled DNA fragments, two DNA uncoiled strands. Now, if you remember, one of them will have polarity 3N to 5N. Other is polarity 5N to 3N. So that strand which has polarity 3 to 5, 3 to 5 upwards, 3 to 5 upwards, which is 3 to 5 upwards, if you remember, the ascending strand. 3 to 5 upwards is the ascending strand. The ascending strand acts as the template, और ascending strand के ऊपर we have nucleotides. Ascending strand has got nucleotides. So sir, समझो here we have G, guanine. Then reading the guanine, an enzyme called as RNA polymerase will bring in new nucleotides and construct an RNA called as the mRNA. If you remember, DNA replication में we had two separated strands. ये था template. And who bought the new nucleotides? DNA polymerase. So DNA replication में DNA polymerase bought the new nucleotides. In RNA formation, RNA polymerase will bring in new nucleotides. RNA polymerase brings new nucleotides. So now check this carefully. You have G, then what will be the complementary mRNA? mRNA के ऊपर आएगा C, and then say you have again G, then you'll have again C, and then suppose you have T, then here will come A, and then you have A, then here will come U, and here you have C, then here will come G, and AUG नाम का एक सीक्वेंस बन जाएगा. Adenine, uracil, guanine. AUG, AUG is something called as a codon, and codon attracts amino acid and begins the process of protein synthesis. And AUG is the first codon which is called as the start codon. AUG is the first codon called as the start codon. So which is the start codon? AUG. And DNA पे कौन सा sequence था? TAC. So T attracts A attracts U. Remember, adenine attracts thymine, but thymine will attract adenine, but adenine will attract not thymine but uracil, because RNA doesn't have thymine, it has uracil. So what will thymine attract? Adenine. What will adenine attract? Uracil. And what will cytosine attract? Guanine. And what is that first sequence? AUG. AUG is called as the start codon. Then triplet on mRNA is called codon. Triplet on mRNA is called codon. Triplet on DNA is called as codogene. So which is the start codogene? Start codogene is TAC on the DNA. And which is the start codon? Start codon is AUG on the mRNA. So DNA पे start codogene is TAC and mRNA पे start codon is AUG. Sometimes TAC is absent, and instead of TAC, CAC is present. Then what will C attract? G. What will A attract? U. What will C attract? G. Then CAC will become start codogene and GUG will become start codon. So start codon is either AUG or it is GUG. AUG or GUG are one of the two start codons, which means which are start codogenes TAC, which codes for AUG, or CAC, which codes for GUG. And these are your start codogenes and start codons. तो ये ट्रिपलेट बन गया. Next sequence on the DNA, say you have again you have CCC. Then on mRNA you will have GGG, and on DNA you have AAA. Then on mRNA you will have UUU. And ऐसे करके triplet, 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 triplet. So what is this triplet called? Codogene. What is this triplet called? Codon. What is this triplet called? Codogene. What is this triplet called? Codon. And approximately 1,000 codons are made. So codogene triplet, codon triplet, codogene triplet, codon triplet, codogene triplet, codon triplet, codogene triplet, codon triplet. And ऐसे 1,000 कोडोजीन्स और यहाँ 1,000 कोडॉन्स के बाद बनता है, आता है एक सीक्वेंस ATT. Then on the mRNA it will form UAA, and UAA is something called as a stop codon. If you remember, AUG was the start codon, and start codon starts protein synthesis. And then we have 1,000 codons which will continue protein synthesis. And then you have UAA, which will be the stop codon. It will stop protein synthesis. As a matter of fact, stop codons are three. You have UAA, or you have UAG, or you have UGA. Any of these three codons will act as a stop codon. So which are the stop codon? UAA or UAG or UGA. तो उसके लिए कॉम्प्लीमेंट्री स्टॉप कोडोजीन्स भी होंगे. Which are the stop codogenes? ATT is the stop codogene which will code for UAA, or ATC will code for UAG, or ACT will code for UGA. And we have ATT for UAG, ATC for ATC for UAG. Sorry, ATT for UAA, ATC for UAG, or ACT for UGA. And that is the way we have start codogenes and stop codogenes helping in the process of transcription. So, which is the start codon? AUG or GUG. Which are the start codogenes? TAC or CAC are the start codogenes on the DNA. And mRNA has start codon AUG or GUG. Start codogenes are TAC or CAC. TAC codes for AUG and CAC codes for GUG, start codon. Then we have codogene codon, codogene codon, codogene codon, codogene codon. Approximately 1,000 codogenes will code for 1,000 codons, and then you will get sequence ATT, which codes for UAA, which is the stop codon. So that is a stop codogene. Or you have ATC, which will code for UAG. Or you will have ACT, which will code for UGA. And ATT for UAA, ATC for UAG, or ACT for UGA. These become the stop codogenes and the stop codon, and thus is produced an mRNA at the C5 end. By the way, ये है C3 end of DNA, and this is C5 end of DNA, which is the ascending strand. So mRNA is formed in which polarity? Opposite polarity. तो mRNA कौन से पोलैरिटी में बनेगा? C5 to C3. DNA is C3 to C5, so mRNA will be C5 to C3. At the C5 end we have start codon, then we have 1,000 codons. At the C3 end we have stop codon, one of the three stop codon. And check this here. ये है आपका C5 end, and here we have AUG. What is AUG called as your start codon? And then we have codon, we have codon, we have codon, we have codon. Approximately 1,000 codons are there. And then at the C3 end, you have UAA, or it could have been UAG, or it could have been UGA. Any of these three come, it is a stop codon. So, which is UAA over here, the stop codon, and what is AUG over here, the start codon. And बीच में approximately 1,000 codons. So this is triplets of nitrogen bases called as codons. At the C5 end start codon, at the C3 end stop codon, बीच में 1,000 codons made with the help of DNA by a process called as transcription. So this is excuse me, this is your mRNA made by transcription. Let's see this now. Working, a sequence of 3 nucleotides on the mRNA is called as a codon. Each triplet code on the mRNA codes for a specific amino acid. Each codon, now check this here, each codon will attract a specific amino acid. तो अभी ये बहुत ध्यान से करते हैं, आपके सेल में क्या होता है, ध्यान से सुनना. सेल में होता है न्यूक्लियस, और न्यूक्लियस के अंदर होता है डीएनए. And what will this DNA do? This DNA, we just said, will undergo a process called as transcription. And DNA forms mRNA by transcription. DNA forms mRNA by transcription. mRNA leaves the nucleus. तो न्यूक्लियस के बाहर आ जाता है and goes into the cytoplasm. And ये है न्यूक्लियस, ये है cytoplasm. mRNA goes into the cytoplasm. Attached to the nucleus is the endoplasmic reticulum. And endoplasmic reticulum has over it structures called as ribosomes. And the mRNA goes and fits into a ribosome. mRNA राइबोसोम के अंदर जाके फिट हो जाता है. राइबोसोम में जाके mRNA फिट हो जाता है. And once the mRNA fits in the ribosome, its codons are read and a message goes into an amino acid pool. तो साइटोप्लाज्म में there is an amino acid pool and amino acid pool has in it different types of amino acids. Amino acid pool has in it different types of amino acid. To be specific, 20 types of amino acids. 20 types of amino acid. You had done that in biomolecule: methionine, arginine, lysine, serine, histidine, glutamic acid, aspartic acid, valine, isoleucine, etc., etc. 20 types of amino acids are there, and the mRNA has got triplets called as codons. Every codon will attract one of the amino acids. Every codon will attract one of the amino acids. And the amino acids are delivered or transferred from the amino acid pool into the ribosome by RNAs, which are called as tRNAs, transfer RNA. तो ये है स्टार्ट कोडॉन AUG और ये है tRNA. This is transfer RNA and it delivers an amino acid into the ribosome. और राइबोसोम में अमाइनो एसिड आता है. Where is tRNA found near the amino acid pool? An amino acid pool has in it how many amino acids? 20 types of amino acid. And tRNA will pick up one amino acid and deliver the amino acid from the cytoplasm into the ribosome, and tRNA will deliver the amino acid to the start codon. So start codon AUG will have one amino acid delivered. Similarly, the second codon is read. It will attract one more amino acid and tRNA will pick up the amino acid and deliver the amino acid into the ribosome. And second codon has one amino acid. Then the third codon is read. It will attract tRNA with another amino acid and form an amino acid group over here. So start codon delivered an amino acid, and second codon delivered an amino acid, third codon delivered an amino acid. और इन अमाइनो एसिड्स के बीच में एक पेप्टाइड बॉन्ड बन जाता है. पेप्टाइड बॉन्ड बन जाता है. How many codons, if you remember, there were approximately 1,000 codons, and 1,000 codons will attract tRNAs with 1,000 amino acids and form amino acid chain by forming peptide bonds. And what is an amino acid chain with peptide bonds called as? A protein. तो ये प्रोटीन बन जाएगा. And that is the way protein synthesis will take place, and that process of codons attracting tRNA, which deliver amino acid from amino acid pool into the ribosome, and ribosome में tRNAs deliver the amino acid, tRNA delivers the amino acid, tRNA delivers the amino acid, and amino acids के बीच में बनता है peptide bond, and peptide bond से बनता है amino acid chain. That process is called as translation. What is that process called as? Translation. So these rules are specific. Check this here carefully. Each triplet codon codes for a specific amino acid. Each codon codes for a specific amino acid, which is called mRNA language or genetic code or cryptogram. mRNA language or genetic code or cryptogram. We will do this in a lot of detail later, but I'm just showing it to you right now. Key, which are the amino acids and what will be the delivery. So, sir, समझो you have codon UUU or UUC. It codes for phenylalanine, and UUA, UUG is for leucine. CUU, CUC, CUA, CUG is for leucine. Similarly, isoleucine. AUG, the other start codon, codes for methionine. AUG codes for methionine. GU series codes for valine, and then you have serine, and then you have proline, you have threonine, you have alanine, you have tyrosine, you have UAA and UAG called as stop codon, and then you have histamine, you have glycine, you have aspartame, lysine, aspartic acid, glutamic acid, cysteine, and you remember UGA is also a stop codon. So UAA, UAG, UGA are all stop codon. AUG is the start codon, codes for methionine. And then CG series codes for arginine. AG series codes for serine. And GG series codes for glycine. तो ऐसे दो तीन याद रखने हैं. AUG codes for methionine. AUG codes for methionine. GGG codes for glycine. GGG codes for glycine. CCC codes for proline. CCC codes for proline. UUU codes for phenylalanine. UUU codes for phenylalanine. So, what does AUG code for methionine? And then you have CCG codes for proline, and you have GGG codes for glycine, UUU codes for phenylalanine. That means a total of 20 amino acids के लिए you have different types of codons. You have codons, triplets, which code for 20 types of amino acids. That language is called as the genetic code. That language of mRNA is called as the mRNA language or genetic code or cryptogram. And that is the mRNA language or genetic code or cryptogram. The codon present at the C5 end of the mRNA is called start codon or initiation codon. Start codon, also called initiation codon. It is usually AUG, sometimes it may be GUG. So that is what we just did. At the C5 end, you have AUG or sometimes you have GUG. What is that called as? The start codon, also called as the initiation codon. And then we have the codon at the C3 end is the stop codon or the nonsense codon. Stop codon, also called nonsense codon. It is either UAA or UAG or UGA. So codon at the C3 end is the stop codon, UAA or UAG or UGA. These are called stop codons. They stop protein synthesis. Also called nonsense codons because they don't code for any amino acid. They don't code for any amino acid. They are called as nonsense codons. And check this here. Each codon on the mRNA specifies a particular amino acid. For example, start codon AUG codes for methionine or F-methionine. AUG always codes for methionine. तो if you remember, हमेशा start codon होता है AUG. And AUG अगर start codon है, तो which is always the first amino acid delivered. अगर ये है AUG, message goes into the amino acid pool. Amino acid pool has 20 types of amino acid, and a tRNA will pick up amino acid and deliver the amino acid into the ribosome. तो राइबोसोम में है mRNA or tRNA, and which is the first amino acid always delivered. For AUG, the first amino acid always delivered is methionine. Methionine is the first amino acid always delivered. So start codon is AUG. First amino acid is always methionine. And then we have other rules. So if it is a eukaryote, then methionine. If it is a bacteria, prokaryote, then methionine के ऊपर एक फॉर्माइल ग्रुप आ जाता है, and that is called as F-methionine. So eukaryotes in us, AUG is for methionine. In prokaryote bacteria, AUG codes for F-methionine, formilated methionine. Similarly, GGG specifies glycine. GGG specifies glycine. Or you have UUU codes for phenylalanine. Or you have CCC codes for proline. Similarly, you have UGU, you have ACG, you have UCA, you have AAA, you have so many codons. Each codon will code for a specific amino acid. Specific amino acid. तो हम लोग दो तीन याद रखते हैं. AUG codes for methionine, CCC codes for proline, UUU codes for phenylalanine, GGG codes for glycine. And let us see what happens. So, sir, समझो this was the beta cell of islet of Langerhans of pancreas. Beta cell of islets of Langerhans of pancreas और बीटा सेल में ये था न्यूक्लियस. न्यूक्लियस में जीन नंबर 11,111 का ट्रांसक्रिप्शन हुआ और बना mRNA. And that mRNA went from the nucleus into the cytoplasm, went towards the endoplasmic reticulum, went into a ribosome, and ribosome में आ गया mRNA, and ribosome में you have mRNA having a specific sequence, and that sequence of codon will attract amino acid from the amino acid pool, and from the amino acid pool you have tRNAs. Amino acid pool may say you have tRNAs transferring amino acid into the ribosome. So let's see what will happen. समझो this is the mRNA. made by transcription of gene number 11,111. Gene number 11,111 is the gene और उसके ट्रांसक्रिप्शन से बना mRNA. ये है राइबोसोम और ये है बीटा सेल. बीटा सेल में है अमाइनो एसिड पूल consisting of 20 types of amino acid. Suppose start codon is AUG. Then tRNA will bring which amino acid? Methionine. Next sequence A is GGG. Then tRNA will bring which amino acid? We have just done that. GGG codes for glycine. Next codon is UUU. Then which amino acid? Phenylalanine. Next codon say is CCC. Then which amino acid, if you remember? Proline. So sir, proline will be the amino acid. And ऐसे ही अमाइनो एसिड्स डिलीवर होते जाएंगे और उनके बीच में बनेगा पेप्टाइड बॉन्ड. And how many codons are coded? If you remember, there are 1,000 codons. 1,000 codons, and 1,000 codons means 1,000 amino acids are added, and then will come stop codon. UAA or UAG or UGA. उससे एक भी अमाइनो एसिड अट्रैक्ट नहीं होता है. And stop codon stops protein synthesis. So, what is the sequence of codons? AUG, GGG, UUU, CCC. What is the sequence of amino acid? Methionine, peptide bond, glycine, peptide bond, phenylalanine, peptide bond, proline, peptide bond, codon, amino acid, codon, amino acid, codon, amino acid. How many codons? 1,000. How many amino acid? 1,000. Then comes stop codon. That means no amino acid transferred. And that means this now translation process has stopped. And this amino acid chain goes from the ribosome into the cytoplasm and is an amino acid chain, a protein. Let us give it a name. This was the beta cell, and this was the beta cell of islets of Langerhans of pancreas. So this protein is called as इंसुलिन. तो इसमें इंसुलिन बन गया. And what is insulin? A protein. What is the sequence of amino acids? Methionine, glycine, phenylalanine, proline, aspartic acid, glutamic acid, glutamic acid, glutamic acid, aspartic acid, serine, 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 phenylalanine, phenylalanine, methionine, methionine, methionine. And ऐसे 1,000 amino acids से बन गया insulin. So which is the first sequence you remember? Methionine peptide bond, glycine peptide bond, phenylalanine peptide bond, proline peptide bond, amino acid ऐसे. How many amino acids? Approximately 1,000 amino acid. इससे बन गया insulin. So sequence of amino acid of insulin is methionine, peptide bond, glycine, peptide bond, phenylalanine, peptide bond, proline, etc., etc. Suppose this is insulin, then what is this mRNA called? mRNA is the cryptogram or mRNA language of insulin. And what was the sequence of codons on it? AUG, GGG, UUU, CCC. तो ये है mRNA जिससे बन गया मीथियोनिन, ग्लाइसिन, फेनिलैलेनाइन, प्रोलीन एक्सेट्रा. By the way, this mRNA is the mRNA for insulin. It was made from DNA. Gene number 11,111. So this DNA से बना mRNA. So DNA has got codogenes. DNA has got codogenes. What is the sequence of codon? AUG, GGG, UUU, CCC, etc. तो डीएनए पे क्या होगा? डीएनए पे AUG के लिए होगा TAC. TAC. TAC is the start codogene. For GGG, what was the sequence present? CCC. For UUU, it was AAA. For CCC, it was GGG. So, what was the sequence of codogenes? TAC, CCC, AAA, GGG. And here is the sequence: TAC, CCC, AAA, GGG, and all the way. So this is the gene for insulin. This is the gene for insulin. What is the sequence of nucleotides in the gene for insulin? TAC, CCC, AAA, GGG is the sequence of nucleotides in the gene for insulin, which forms mRNA having AUG, GGG, UUU, CCC, which forms amino acid methionine, glycine, phenylalanine, proline, etc. with peptide bonds, and that is insulin. So, methionine, glycine, phenylalanine, proline, etc. is the amino acid chain called insulin. So AUG, GGG, UUU, CCC is the mRNA language for insulin or mRNA for insulin, which means TAC, CCC, AAA, GGG, etc. is the codogenes for insulin, and that is sequence number 11,111 is the gene for insulin. So gene number 11,111 has undergone transcription to form mRNA, and mRNA has undergone translation to form a protein. Suppose this was the beta cell. Suppose this was muscle cell. मसल सेल के अंदर सेल, सेल के अंदर न्यूक्लियस, न्यूक्लियस के अंदर डीएनए, एंड डीएनए में जीन नंबर 11,111. No. That is for insulin. Gene number 11,111 will not undergo transcription, translation and muscle. क्योंकि वो तो जीन फॉर इंसुलिन है, वो सिर्फ बीटा सेल में ही ट्रांसक्रिप्शन ट्रांसलेशन करेगा. मसल में डीएनए तो सेम है. The all DNAs have come from same zygote, but gene number 2683. Gene number 2683, DNA uncoils by breaking of hydrogen bond. It forms mRNA by transcription. It forms amino acid chain. Suppose the sequence of codons is AUG. That is always the golden rule. And then you had GCG, you had CGG, you had AGA, you had CGC, different sequence. For इंसुलिन क्या था? इंसुलिन के लिए था AUG, GGG, UUU, CCC. Now for gene number 2683, the sequence is different. And it will form amino acid chain methionine, serine, glycine, serine, proline. So what is the sequence? Methionine is always constant. And then you had serine, glycine, serine, proline, and will form a chain of amino acid and start to stop codon. एक amino acid का sequence बनेगा. That protein is released. What is that protein called as? Actin and myosin. ये मसल सेल था तो ये मसल प्रोटीन बन गया. And what was the sequence of amino acid in insulin? In insulin, it was methionine, glycine, phenylalanine, proline, etc. What was it in actin myosin? It was methionine, serine, lysine, serine, proline, etc. Sequence of amino acid alag tha, which means sequence of codons was different, which means sequence of codogenes was different. Sequence of codogenes, in other words, TAC और उसके बाद था some other sequence on DNA, उसके बाद कोई other sequence on DNA, उसके बाद कोई other sequence on DNA, and that means DNA has got another sequence, which means it's a different gene. Similarly, what will happen in, say, hair cell, in your skin cell, उसके अंदर gene number 22,683, 22,683 undergoes transcription to form mRNA. उसके ऊपर कोडोजीन sequence of nucleotides is different. उससे बना mRNA with start codon AUG, stop codon UAA, UAG, UGA, but बीच में sequence of codons was different. उससे बन गया amino acid sequence methionine, isoleucine, 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 serine, 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 aspartine, 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 lysine, histidine, 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 और ये बन गया keratin, which forms your hair. And say gene number 5181 in your bone cells undergoes transcription to form mRNA and translation to form amino acid chain. उसमें sequence है methionine, glycine, serine, leucine, isoleucine, phenylalanine, 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 valine, 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 threonine, 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 और उससे बन गया अमाइनो एसिड सीक्वेंस ये वाला, which will form another protein called ossin, that is bone protein. And if you have understood sequences of amino acid, 20 types of amino acid sequences are different from each other, and 20 types of amino acid sequences are different. एक सीक्वेंस से बनता है इंसुलिन, एक सीक्वेंस से बनता है अलग, another sequence forms of amino acid forms actin, myosin, some other sequence forms keratin, some other sequence forms ossin, some other sequence forms growth hormone, hormones are proteins, some other sequence forms hexokinase, enzymes are protein, some other sequence forms isocitrate dehydrogenase, which is enzyme, which is protein, some other sequence forms prolactin, which is a protein, some other sequence forms odontoblast and ameloblast cell के proteins which form your teeth, and अलग अलग amino acid sequences से अलग अलग genes, अलग अलग proteins बनते हैं. By the way, all proteins from head to toe, we are nothing but walking, talking bags of proteins. Hair keratin, skin collagen, collagen, muscles actin, myosin, cartilages chondrin, bones ossin, Enzymes are proteins, hormones are proteins. All proteins are nothing but permutation combination of 20 types of amino acid, which means they all are different depending on the sequence of codons on the mRNA. क्योंकि codons of mRNA decide sequence of amino acid, which means all are different depending on codons of mRNA, which means they all are different depending on the codogenes of the DNA. And DNA के ऊपर अलग अलग sequences of triplets called codogenes decide sequence of codon, and the sequence of codon on mRNA decide sequence of amino acid in the protein. So gene number 1 undergoes transcription translation to form a different protein. Gene number 2 undergoes transcription translation to form a different protein. ऐसे 30,000 genes होते हैं. And if you all have understood, from the first cell called zygote, it formed 3 layers: ectoderm, mesoderm, endoderm. ectoderm के अंदर जीन नंबर से 11,968 underwent transcription translation to form proteins which form the brain. जीन नंबर mesoderm के अंदर जीन नंबर 2,638 underwent transcription translation to form actin myosin, which means mesoderm से बना muscles. Some other gene number 5,238 underwent transcription translation to form ossin, जिससे mesoderm से बना bone. Gene number 28,238 से बना chondrin, which forms cartilages. And endoderm से gene number 263 से बना कनेक्टिव टिश्यू, जिससे बना लंग्स के एल्वियोलाई के साथ कनेक्टिव टिश्यू से बना रेस्पिरेटरी सिस्टम. Everything was made by different transcriptions and translations of different types of proteins, of amino acids. And all proteins from head to toe are permutation combination of 20 types of amino acid, which depends on the permutation combination of nitrogen bases on the mRNA, which depends on the permutation combination of nitrogen bases on the DNA. So, 4 alphabets: adenine, guanine, thymine, and cytosine on the DNA. 4 alphabets के अलग अलग परम्यूटेशन कॉम्बिनेशन से बनते हैं कोडोजीन्स, उससे बनते हैं अलग अलग कोडॉन्स, उससे बनते हैं अलग अलग सीक्वेंसेस ऑफ अमाइनो एसिड, which means the entire human body is made from nothing but different combinations of 4 alphabets: adenine, guanine, thymine, and cytosine on the DNA. What a beautiful logic. I hope we all have understood. This was the mRNA.